announcements. This is, maybe this is a trial run for the way online education in this course will be after spring break. We'll see. It all depends on things outside of our control. Be ready to use Moodle and email. We'll figure it out. On Thursday, assuming everything is as it is today, Lindsay will be giving a science talk about. So my understanding is now the virus is called SARS-CoV-19 or something. SARS-CoV-2. SARS-CoV-2 is the name of the virus, and the name of the disease is COVID-19. That seems to be where the field is going. Anyway, she's going to be talking about the viral mechanisms and what her knowledge about the virus and the epidemiology thereof. So that will be upstairs in the 300 on Thursday, assuming we're still having gatherings like this. Let's see, what else? I want to make an announcement about a 7-week AWPE course that's available here. If you want AWPE credit or want to just get some exercise outside and do some useful work out in the woods, Brent Smith is leading an AWPE class in trail building and invasive species control. Basically get out in the woods and destroy honeysuckle and build trails. Honeysuckle is an invasive species that woods people hate. It's a nice little plant, but it's invasive and it's really nasty out there. So if you want to do this, there's some things to do. It's starting this week, I guess. Register now, meet outside the CoLab this week, 1 o'clock. Here's the email for more information. I have a thank you note for the people at the butcher shop, for the slaughterhouse where we get our eyes, the pig eyes, or the cow eyes. And so, but since, I think what I'll do is help me remember in lab today and on Thursday that I want to circulate this thank you card for you to sign to go to the workers. They work for us for free, and so we send them a thank you. Okay, so today we're going to move from sensory input into motor output. We receive information from the world, we process it, sometimes we remember it and sometimes we don't. Sometimes we think about it and sometimes we don't. Did you get the slides? But in any case, so we're not going to talk about cognition in this course, thinking about it, emoting about it, feeling about it, remembering it. That's not what we're going to talk about in this course. We're going to talk about the output, your body's response, the motor system. So we'll talk today mostly about muscle. Some of this will be review from CellPhys, and some of this will be new. That's good. And we'll get an overview of the motor system, not just muscle, but the way your nervous system controls your body, your muscles. So that's kind of where we're going today. Muscles, type of muscles, and then neuromuscular processing. So here is an overview of the motor system. So here's the output unit, right? We move our muscles. That moves our body. And of course, here is the input. The highest level in the motor system is the motor cortex. And you'll see that the motor cortex is in the precentral gyrus, right next to the somatosensory cortex. So it's in the most posterior gyrus of the frontal lobe, the precentral gyrus. So that's, the motor cortex is where, it's not where it begins, but it's where we have our highest level processing of motor planning. The muscles are where the output is. And there's lots of interesting things in between. There's a spinal circuit. This is a local, circuit that does a lot of the planning and organization of the movement. So we have somatosensory input in the form of proprioception, processing right here in the spinal cord, and output even without any brain. And in addition to the cortex, we're going to have input from the basal ganglia. Remember that's the caudate, the putamen, and the globus pallidus, along with the substantia nigra. We're going to have some input from more core centers of the brain into the motor cortex, as well as from the cerebellum, output from the cortex that drives these central pattern generators. That's what these are. CPGs, central pattern generators that sort of control stereotyped movement. You'll see what I mean in a minute. So we're going to focus today here and then work our way back upstream. Okay, muscle. You probably know that there are three main types of muscle: cardiac muscle, smooth muscle, and skeletal muscle. We're focusing on skeletal muscle. We talk about cardiac and smooth in AMP2. So skeletal muscle is, well, you've seen it in the lab now. Now we're sort of catching physiology up with the anatomy that you've seen in the lab. Skeletal muscle is a big bundle of muscle fibers connected by a tendon, usually to a bone, but not always to a bone, sometimes to skin, or sometimes to something else. But typically, it's muscle, tendon, bone. The muscles are comprised of muscle fibers. Muscle fibers are the cell of muscle. They're big, polynucleated, so these dots are supposed to represent the nuclei of a single fiber. They're big cells because they're formed by the fusion of embryonic or other stem cells. So stem cells fuse to form these big muscle cells called muscle fibers, and these muscle fibers bundle together to form a muscle. So myoblasts and myotubes are the stem cell precursors to muscle fibers. So zooming in on a muscle fiber, this is zooming in. A muscle fiber has a nucleus. It actually has scores of nuclei. It's polynucleated. And within each muscle fiber are myofibrils. These are the contractile unit. Well, myofibrils make up muscle fibers, which make up muscles. Right? That's, we're zooming in. And each myofibril is comprised of a stack of sarcomeres. Thousands of them. Thousands of sarcomeres connected in series. And each sarcomere has the proteins, the unit for contraction. So each sarcomere is going to get shorter, during contraction, which means the muscle fiber gets shorter, which, if they're all working together, the muscle gets shorter, and that's muscle contraction. So muscle, at its most basic, at a basic cellular unit, before we get into molecules, the contraction of a muscle is due to the shortening of sarcomeres. A few more words of muscle anatomy that we haven't used in the lab. A muscle is wrapped by that shiny layer that you've seen in the lab that surrounds a muscle has a name, and it's called the epimysium. The the epimysium is kind of continuous with the tendon. So the tendon and the epimysium are the same sort of connective tissue, collagen mostly. Epimysium surrounds the muscle, and then where there's no more muscle and it's connected to the bone is the tendon. Within each muscle, of course, there are muscle fibers. That's what I just told you before. But a bundle of muscle fibers within the muscle is called a fascicle. A fascicle is a bundle of muscle fibers within the muscle at large. And each fascicle is surrounded by perimysium, a membrane, extracellular matrix. So, muscle fiber, fascicle surrounded by perimysium. Each muscle fiber is surrounded by an endomysium, and then each muscle is surrounded by an epimysium. I might quiz you on one or more of these details with regard to muscle anatomy. So there are three main types of muscle, of skeletal muscles. And you may be aware of them. And those of you who are athletes are probably already aware of them. So let's just go through them. There are the slow muscles, the fast fatigue-resistant muscles, and the fast fatigable muscles. So this is in order of speed. The slow muscles are red in color, darker in color than the fast ones. They have when I say they have slow myosin, they have an isoform of myosin that is slow to do its contractile processing relative to the other two. So it contracts slowly. They have lots of mitochondria, so they get their ATP from what you learned about in self-phys, oxidative phosphorylation. Almost all of their ATP is from mitochondrial oxidative phosphorylation. Glycolysis, Krebs cycle, electron transport chain, ATP synthase, oxidative phosphorylation, oxidative production of ATP. They're red because they have lots of mitochondria that are brown, and they have lots of myoglobin, which is red. Myoglobin is an oxygen-storing protein similar to hemoglobin in the blood. So red muscles, slow twitch muscles, are muscles that we use kind of all the time. They're postural muscles. So, I have evolved to stand upright, so muscles in my legs and my hip are contracting all the time that I'm standing. I don't get tired of it. I could stand, I will eventually get tired of standing, but I can stand for a long time because I'm using slow twitch muscles that are producing ATP, plenty of ATP. As long as they get blood in order to get oxygen and nutrients, they can keep up with the ATP production, but they don't contract very fast. They contract regularly. They're good at holding a posture. So let's jump from the slowest to the fastest. The fastest are the fast twitch muscles that are fatigable. These have the fastest myosin, so the isoform of myosin there is the fastest in doing the cross-bridge cycling that we'll get to in a minute. They don't have very many mitochondria, so they produce most of their ATP by glycolysis, which reflects why they're fatigable, because they can produce ATP like that, but not for very long. ATP production by glycolysis. So for example, what's an example of a muscle that you're aware of that are fast twitch? The the muscles that control your eyelids. They twitch very fast. You can blink very quickly. But if I were to put a weight on your eyelashes and ask you to try to raise it with your eyelids, you couldn't do it because they're not very strong. There's not very many muscle fibers. But they would get fatigued very quickly if they were trying to lift any weight other than the eyelashes. So these are very fast, but very easily fatigable. And that's why when there is a disease of musculature, these muscles, the eye muscles, not just the eyelids, but the superior and inferior rectus, the lateral and medial rectus, the muscles that move your eyes, they don't have to lift any weight, but they have to move very quickly. So the eye muscles are in this category, and they are some of the first muscles to show defects in a neuromuscular disease. So for example, there's a disease called myasthenia gravis, which is an autoimmune disorder of the neuromuscular junction, and the first symptom of myasthenia gravis is drooping eyelids. In between slow, postural, and fatigable fast twitch muscles are the fatigue-resistant fast twitch muscles. So they have an intermediate speed myosin. They do both glycolysis and mitochondria. They produce ATP by glycolysis and by oxidative phosphorylation. They are fast, but they're fatigue resistant because of the mitochondrial production of ATP. So they're fast, but they don't fatigue as fast as these muscles do. So they're in between. I will not require you to learn the nomenclature, the type 1, type 2A, and type 2B nomenclature. Just remember that there are the three types: slow, fast, fatigable, and fast, fatigue resistant. And know what the difference is between them. Yeah?
Are there one that's like more efficient?
More efficient. I think it depends on your definition of efficiency, and I don't know the answer to that. Most of your muscles are a mixture of slow, fast, and fast fatigable, and fast fatigue resistant. Most of your muscles are a mixture of those. My gluteus maximus are mostly slow. My eyelid muscles are mostly fast, almost exclusively fast fatigable. My biceps and my rectus femoris are intermediate. They're mixed. Saw a hand up over there. Remy?
Yeah, I was going to ask for fatigue resistant. Where do we see that?
We see that almost everywhere. So fast fatigue resistant is mixed into almost all of your muscles. Except for the ones that are strictly postural. Like, the gluteus maximus is used for running, so I'm trying to think if there's a muscle that is only used for just standing. The lower back muscles? Yeah, okay. So these are generally fatigue-resistant muscles. Let's see, what are some of these muscles? The erector spine. The three muscles of the erector spine. Let's see, can I remember what they are? Iliocostalis, longissimus, whatever, and spinalis. Did that answer your question?
Yeah. You were saying it's muscles that aren't related to posture?
Muscles that you use to move are usually a mixture, because sometimes you need to move slowly, and sometimes you need to move fast. Right? So that they are usually a mixture. But the muscles that you use for posture only, and there's only a few of them really, are mostly slow. And the muscles that you only use to move fast, like your eyes, and don't ever have to support any weight for any length of time, are mostly fast, fatigable. Any other questions? So each muscle, so this is an image of some muscle, of a muscle, with muscle fibers. Each of these red stripes is a muscle fiber. And each of these sort of oval dark things is the synapse of the motor neuron that innervates a single muscle fiber. So here's the nerve that branches out and forms synapses with these 1, 2, 3, 4 muscle fibers. One nerve, 4 muscle fibers. This one appears to not be innervated by this nerve. It's presumably innervated by some other nerve, some other motor neuron. So that defines, we get to a thing called the motor unit. A motor unit, so each muscle fiber is innervated, each muscle fiber has one neuromuscular synapse from one motor neuron. So one motor neuron innervates and triggers the contraction of one muscle fiber. A group of muscle fibers can be innervated by one motor neuron. So that's what we see here. A group of muscle fibers innervated by one motor neuron. That's the motor unit. The group of muscles innervated by one motor neuron. So each muscle fiber is innervated by one motor neuron, but each motor neuron can innervate many muscle fibers, and that's the motor unit. So this is an image of one motor unit, and here's an image, helpfully colored, of 2 motor units in the same muscle. So each muscle might have hundreds, maybe even thousands, of motor units, because they, you know, if they have 10,000 muscle fibers, and they're innervated by 1,000 motor neurons, then on average, each muscle unit had 10 muscle fibers in it. Is that clear? So now we're going to get into some material that is a review of cell-phys. Okay, I'm going to go through it, but this is going to be reviewed. So the synapse. Well, let me go back. Well, what I'm zooming in on here is the synapse between the motor neuron and the muscle cell. This is what I'm zooming in on. It's mostly in a cartoon version. Okay, this is an electron. This is not a cartoon. This is an electron micrograph of that synapse. Here's the presynaptic nerve terminal. Here is the muscle cell membrane. Here is the synaptic cleft. Here are the synaptic vesicles containing acetylcholine, because that's the neurotransmitter at the neuromuscular synapse. So the neuromuscular synapse is also called the neuromuscular junction. It's completely interchangeable terminology. And I'll usually abbreviate it NMJ. So that's the synapse between the motor neuron and the muscle fiber. Acetylcholine is the neurotransmitter at the neuromuscular junction in mammals. It's not the neurotransmitter in insects. We're not insects. Acetylcholine is the neurotransmitter. So there's, vesicular release. You remember the story. Acetylcholine release, diffusion, and it interacts with acetylcholine receptors, nicotinic acetylcholine receptors, which are ionotropic, cation permeable ion channels that are opened by acetylcholine, and that's the receptor here. There is this stereotype structure of so-called post-junctional folds. It's what happens at the neuromuscular junction. And we'll see that again in cartoon. It's stereotyped, it's just there. It increases the surface area of the muscle underneath the nerve at the synapse. Okay, so neuromuscular junction. So let's go through the story of the neuromuscular junction. Well, actually, why don't I do this as a think, pair, share. I want you to work with the person next to you or around you. So let me get you oriented. Here's the motor neuron. Here's the axon of the motor neuron. Here's myelin of the Schwann cells that myelinate the motor neuron. Here's the presynaptic terminal. It has many mitochondria, only one of which is cartooned here. It has many more than this of synaptic vesicles. Here's the synaptic cleft, which is exaggerated in distance. Here's the muscle membrane with the post-junctional folds, which are exaggerated in their sharpness. The acetylcholine receptors are located at the top of the junctional folds. And voltage-gated sodium channels are located at the bottom of the junctional folds and along the part of the membrane that is not at the motor junction, at the synapse itself. So the muscle membrane is covered with voltage-gated sodium channels, and they are expressed in the junctional folds. So what I want you to do with your partner is tell me the story. Start with an action potential in the axon, write out the story of what happens until we get an action potential in the muscle. Okay, let's take 5 minutes for you to work on that. I gave you a box, it's probably too small, but you have extra room on your piece of paper. So we start with an action potential in the nerve. Okay, what's the next step? Let's go around the room, actually. Okay, we'll start. Alexandra, what's the next step? Okay, voltage gated. Can you see if I write here? No. Can you see if I? Okay. Well, voltage gated calcium channels open. Where are they? They're located in the presynaptic terminal. Okay. What's next? Ari. Calcium inflows into the presynaptic terminal. Okay. What's next?
Calcium binds to synaptic.
So calcium binds to synaptotachnum. Okay. Pablo? Endocytosis? No, we've already done all the endocytosis we need. Okay.
I think conformational change
Yeah.
of this name.
Conformational change. That leads, give me one more thing. What conformational change of what?
This name.
Of the snare complex, of T snares and V snares. Okay, Sophia?
Then there's gonna be fusion of.
Fusion. There it is. All you have to do is give me one word. Well, I mean, sometimes they need more than one word. Fusion of what? The vesicles, fusion of vesicles with the membrane of the nerve. Okay? Good. I'm just reviewing the story that from the beginning of the semester that I want you to still have in your head. Okay, Elena. We've got fusion of the vesicles. Okay, release. I'll use your word, but there's a word that I'm looking for actually that's not release. Release of ACH via fusion and what's the word I'm looking for? Begins with a D, diffusion. Diffusion of acetylcholine in the synaptic cleft. Okay, Abby? Okay, right. ACH binds to nicotinic acetylcholine receptors on the muscle membrane, on the tops of the junctional folds of the synapse, here. Okay, now home? No. Tegan? Okay, I'm gonna voltage-gated sodium channels open, but there are things in between. Jennifer? Matt?
So that creates an excitatory postsynaptic.
Okay, EPSP in the muscle membrane, but I want to tell me what causes the EPSP at a sort of molecular level. Why is it E?
Is it
CJ?
because it's acetylcholine and that's an excitatory neurotransmitter?
That's not a that's not a wrong answer, but it's not a good answer.
Okay.
You have an idea, Sheldon? Why does ACH binding to nicotinic acetylcholine receptors cause an EPSP? That's what I'm getting at. What are nicotinic acetylcholine receptors, and what happens to them when ACH binds to them? Charlie?
Influx of cations.
Yeah, they open, they are ionotropic receptors. EPSP is caused by sodium influx, right? That we're getting all the way back to the ions that I want you to not have forgotten about. Sodium influx causes an EPSP. The EPSP is greater than, EPSP is greater than threshold. We don't need to summate this one. This is one where one EPSP is big enough to bring the cell to threshold. Voltage-gated sodium channels open. Are we done? Pretty close. I mean, voltage-gated sodium channels open here. What does that cause here? Where are we at? Remy, do you know what does the opening of voltage-gated sodium channels cause?
Influx of
Okay,
sodium.
fine, you're right, influx of sodium. Ashley?
Depolarization.
Okay. Now I'm making you think too hard. Now you're overthinking. All I'm getting at is there is an action potential in the muscle fiber, right? You knew what I mean, you just didn't know, you were trying to read my mind. The sodium influx, the depolarization through voltage-gated sodium channels triggers an action potential, but now we're in an action potential in the muscle. And this action potential propagates along the muscle, a meter in some cases, in the case of a long, big muscle, all across the muscle. Okay, so here is my answer. It's either already on or will be on Moodle. So you have my answer. Action potentials in the neuron, depolarization, voltage-gated calcium channels, calcium influx, calcium binds to synaptotagmin, conformational change, release of ACH, we're doing pretty good, release of ACH. I forgot to put diffusion, but it's there. ACH binds to nicotinic receptors, receptors open, influx of sodium, depolarization, voltage-gated sodium channels, more depolarization because it reached threshold, action potentials in the muscle, action potential propagation along the muscle membrane. That's the story of the neuromuscular junction. Questions about that? So now I want to remind you of stuff that you learned from Cell Phys. This is called excitation-contraction coupling. How does excitation, that is to say action potentials in the muscle, lead to contraction of the muscle? Let's walk through this. Here is the action potential. Remember, a big transient depolarization of the membrane potential of the muscle. And it is traveling along the muscle membrane. The skeletal muscle is perforated, not perforated because it's not holes, but it's got deep infoldings called T-tubules. In fact, I've got a model in the lab which I will bring after break. So I forgot to bring it this morning, but I will bring it after break. Help me remember that there's a big, blow up of a skeletal muscle that will show you this network of T-tubules. Inside the muscle cell is this network of endoplasmic reticulum called the sarcoplasmic reticulum because it's muscle. So this is a specialized endoplasmic reticulum called the sarcoplasmic reticulum. And you'll remember that that's where the circa, this is circa. Circa keeps the calcium concentration in the sarcoplasmic reticulum very high. Pumps it out of the cytoplasm into the lumen of this SR. This is circa. So I'm just setting it up now. We've set up a membrane, T-tubule, network of sarcoplasmic reticulum, and here is the sarcomere that's going to contract. So this is setting it up. Now let's trigger an action potential. An action potential, a depolarization of the membrane, travels into T-tubules. It's just illustrating one, but they're all, thousands of them, are getting depolarized. In the T-tubule of skeletal muscle, there is a specialized contact between a protein, this is the T-tubule membrane, so this is zooming in here. This is the T-tubule membrane, and this is the SR membrane. And this is cytoplasm in between, microscopically thin, nanoscopically thin. So this is the T-tubule membrane, this is the sarcoplasmic reticulum. The membrane of the T-tubule has now it gets a little fuzzy. It has a voltage-gated calcium channel. But it doesn't function as a voltage-gated calcium channel. It just functions as a voltage-gated. It's a voltage sensor. It detects the depolarization, undergoes a conformational change. Physically connected, that conformational change causes the opening of another calcium channel that is in the SR membrane. And I call this the calcium release channel, because that's what it does. It releases calcium from the SR. So the names here from your book and from lots of sources call this the dihydropyridine receptor because there are drugs. So this is a pharmacological name. There are drugs called dihydropyridines that some of your grandparents are taking now for heart disease or high blood pressure. There are drugs called dihydropyridines that block this receptor, that block this channel. And so a pharmacologist calls this protein the DHP receptor. So this is a pharmacological name for what I call the voltage sensor. There are toxins called ryanodine that block the calcium release channel. So that a pharmacologist calls this the ryanodine receptor, and I call it the calcium release channel. I will accept either name, because you'll see in your book it's called DHP receptor and ryanodine receptor. Fine. If you want to be a pharmacologist, which I am, you can call it those names. But if you want to be a physiologist, you can call it the voltage sensor, and the calcium release channel. Okay, let's not worry about the name, those are the names. What happens is the action potential causes a conformational change in the voltage sensor. That conformational change is communicated to the calcium release channel that opens, and calcium is dumped through this channel from the SR into the cytoplasm. Remember, the calcium concentration was high inside the SR because Circa pumped it there. So you could say, well, wait a second. We've got a channel that's releasing calcium and Circa that's pumping it back in. Who wins? Well, between contractions, Circa wins, pumps the calcium, and allows the muscle to relax. During contraction, this dumping wins, and we get calcium-dependent contraction. If your muscle is contracting continuously, this is sort of a steady state of calcium release and calcium pumping. Clear? So, calcium, which triggered acetylcholine release in the presynaptic terminal, calcium triggered presynaptic neurotransmitter release, it also triggers contraction of the myofibrils. We haven't gotten there yet, but that's where we're going.
Can you just say that about contraction one more time?
Calcium,
When it contracts, will it?
we haven't gotten to the mechanism of contraction. Let's do that and then we'll take a break. Calcium is going to bind to a protein on the thin, so now I've got to get to the next slide. The myofibrils that are made up by sarcomeres are formed of two main proteins, two main structures. Thin filaments that are made up of actin and thick filaments that are made up of myosin. The thin filaments are polymers of actin and a calcium-binding protein called troponin. Actin and troponin are the thin filament. The thick filaments are myosin, and you might, hopefully you remember from cell phys that myosin are motor proteins. Myosin, this is myosin that motors along actin when allowed to do so. The myosin heads, which are shown as these little chevrons here, the myosin heads are making contact between a thick filament and a thin filament. So let's take a break, because now it gets into the importance of calcium and how that triggers contraction. Take a break, I'll go get my muscle model, and we'll turn calcium into contraction. Calcium inside the muscle into contraction. Let's take a short break. Muscle fiber. Let me see if I can decode it for you. Pink are the myofibrils. These are the sarcomeres that do the contraction. So here, this is a sarcomere. This is a myofibril, and this is another myofibril. And you can see in cross-section a bunch of myofibrils. This is the motor neuron, myelinated motor neuron, and here is the motor neuromuscular junction. Neuromuscular junction, motor neuron, myofibrils. The blue network are the T-tubules. And the peach-colored network are the SR. And the little things with squiggles on the middle of them are mitochondria. So let's pass this around. So what I want you to see is the network of T-tubules in blue and the meshwork of sarcoplasmic reticulum in peachish color. So while that's working its way around, let's talk about how the myofibrils get shorter and contract. Remember, we have released calcium from the SR. So cytoplasmic calcium concentration is high. Calcium binds to troponin on the thin filaments and causes a conformational change. When calcium binds to troponin on the thin filament, a myosin binding site on the thin filament is exposed, and myosin heads bind to the thin filament, bind to the actin. So calcium, troponin, conformational change, myosin-actin interaction. That's what we're doing now. Actin with troponin. Myosin as a bundle of myosins, each of which has a head. When calcium is not bound, low calcium concentration, the binding site on actin for myosin is blocked and there's no contraction going on. In high calcium, troponin conformational change moves tropomyosin, exposes the binding site, allows myosin to bind, and now we're going to get into that cross-bridge cycle that you learned about in cell phase. So let's start at number 1 here. Calcium moves troponin and tropomyosin so that myosin can bind to actin. Here it can't, and here it can. Myosin is an ATPase. It hydrolyzes ATP. You want me to move that around?
Oh, no, there's no question.
Okay.
I don't know.
No, blue is the network of T tubule. So blue is contiguous with the surface membrane, even though it's a different color. So blue is T tubule, peach is SR, sarcoplasmic reticulum. There's no, I don't see any blood vessels, but there are some. Myosin is an ATPase. It binds ATP and hydrolyzes it. It's an ATP-dependent motor. So from the now it's a cycle. So at the end of the previous cycle, ATP was hydrolyzed, and ADP and phosphate were bound to the myosin. This is the ADP and phosphate were from the previous cycle. When myosin binds to actin, it undergoes a conformational change, lowers the affinity for ADP and phosphate, which dissociate. And ADP and phosphate dissociation causes another conformational change of the myosin, which causes it to undergo a conformational change from an extended conformation to a flexed conformation. So what just happened? The myosin was bound to actin, and it flexed. And that pulled actin. That pulled actin in this direction. On the other end of the sarcomere, the same thing is happening. So this is myosin pulling on actin here, and over here, myosin is pulling on actin this way, Why is that? Because the myosins are bundled in this tail-to-tail version. So the heads of this one are sort of reaching out this way, and the heads of this one are reaching out this way. And so when I go from extended to flexed, I have pulled this way, and I have shortened the sarcomere. Clear? So what we have just done is pulled actin this way, on this side. Pulled actin this way on this side. Okay, one of these pulls doesn't do us any good. We need to do this 100 times in order to shorten the muscle. So we need the cycle. ATP binds to myosin. ATP binding to myosin causes it to release from actin. So another conformational change. ATP binding to myosin releases the myosin from actin. It's still in the flexed conformation. ATP hydrolysis, which is what myosin does, causes it to extend to the extended conformation. So now we're back where we started from, except that the actin is now in a different position. So now this myosin is going to bind to the actin in a place that's closer to the end of the sarcomere. So we have moved the sarcomere, we have shortened the sarcomere. I need 2 volunteers. Matt? Okay, Matt's going to be a thin filament. This is appropriate. We need another thin volunteer. Who's a tall, thin volunteer? Sophie, you're tall. Come on, you can be another volunteer, quote-unquote volunteer. I just volunteered Sophie. Okay, so I am the thick filament. I am myosin. You are actin. I am myosin. No, you have to get further away because you are thin filaments. So there's a thin filament, and there's a thin filament. Move, please stretch. We need to stretch this muscle a little bit. Okay, a little bit more. Okay, we're relaxed. This muscle can be stretched, just like we just did, right? The myosin and the actin can move back and forth because the myosin is not attached to the actin. So some other muscle can stretch this muscle. We can't contract it until calcium comes in. So, okay, so let's build our sarcomere. Okay, thin filament, I'm the thick filament with my myosin. Okay? No, low calcium. So this muscle could be stretched, right? Because I'm not attached. My myosin is in the ADP and phosphate bound form from the previous cycle. Okay, calcium comes in, conformational change of the troponin and tropomyosin that you can't see, but it's on the thin filaments. I bind. Okay, binding causes a conformational change that causes the ADP and phosphate to be released. There you go. No, keep your fibers nice and straight. Just move just move a little closer, okay? So let's try that again. Okay, so all I want you to do is slide this way. Don't move your arm. Just slide this way. Okay? So I do my conformational change. ADP and phosphate releases. I go into the flexed configuration. That is the power stroke. ATP binds. No, stay where you are. Because there are others of me holding them in place. Thousands of me's holding them in place. ATP binds. ATP is hydrolyzed. I bind. ADP and phosphate are released. Good. ATP binds. ADP and phosphate are released. Let's see. ATP binds. ATP is hydrolyzed. Binding. ADP and phosphate are released. Power stroke. See what just happened? This sarcomere is getting shorter because of this cycle of motoring. My myosins are motoring along their actins, bringing them together. I don't move, at least not if I'm at the center of this thing. Thank you very much. The sarcomeres got shorter. When we're dead, we're not breathing, oxidative phosphorylation stops, production of ATP stops, Circa doesn't pump calcium anymore, so calcium goes up. Calcium causes the contraction, but there's no more ATP, so we get into the contracted state, but we can't let go. We being myosin. We being myosin. The myosin can't let go of actin. The muscle has contracted and stays contracted. That's rigor mortis, because there's no more ATP. Yeah?
Would the opposite of that be like being tased?
Tased. No, tasing is really this. So, I mean, my understanding, I've never been tased, I hope to never be tased, but my understanding is that tasing activates the muscle and causes it to contract, and so you go into sort of a spasm. So it's not the opposite of this, it's a simulation of this. Okay, so we have contracted muscle. So now let's do some muscle jargon. You may have noticed on one of the first slides, I gave names to these structures. I skipped over it there, but now I want you to know about it. So here's the thick filament. I'm the thick filament. My body is the M line, where the middle of the thick filament is called the M line. Matt and Sophie were the Z lines, and their arms were the thin filaments. From Z line to Z line is a sarcomere. So you can see here, if the thick filaments are pulling on the thin filaments, we're pulling this Z line this way, and we're pulling this Z line this way, the sarcomere is getting shorter. Remember, Matt was a Z line, and Sophie was a Z line. Their arms were the thin filaments. My arms were the thick filaments. And if this thick filament is pulling this, if this thick filament is pulling these things this way, and this thick filament is pulling these thin filaments this way, the sarcomere, Z line to Z line, has gotten shorter. And you do that 1,000 times, stack these 1,000 times, and the muscle got shorter. Each sarcomere might shorten by 10 microns. That's a number off, I'm just pulling that on my head, 10 micrometers. But if I have 1,000 of them lined up, then the muscle has shortened by 10 millimeters. And that's how you get muscle contraction, because 10 millimeters is a centimeter. Obviously, muscles can shorten by more than a centimeter, but you see where I'm going. So the M line is the middle of the sarcomere. The Z lines are the end of the sarcomere. Where the thick filament and thin filaments overlap is called the A band. Well, basically, the length of the thick filament is the A band. In the middle of the A band, where there is no thin filament, is called the H zone. And where the Z line is, but there is no thick filament, is called the I band. So Z line, I band, A band with an H zone in the middle, and then repeat. I, Z. Names for the structure of the sarcomere. So I may or may not ask you to reproduce this, but I will use this language when if I talk about a Z line, I expect you to know what that is. I might ask you what defines the end of each sarcomere, and you'd have to come up with Z line. So how do I remember this naming? Well, 10 years ago, there was a student in this class who came up with a mnemonic that I think is pretty good. I'm sure there are other mnemonics. Hers was, Zeus is a heroic, let me do it again. Zeus is a heroic man. It's wrong, right? Because Zeus is not a man at all. Zeus is a god. But you see,
He's also not really heroic.
and he's not heroic either. But so it's a bad mnemonic. But you know, come up with your own. After 10 years, I'm willing to hear another one. So that defines half of this argument. And then it's a palindrome. You do it in the opposite direction. Man, a, okay. Man, H, whatever. You do it in one direction and then you do it in the opposite direction. Once you get the letters, you can do it in a palindrome. What happens is let's get back to the physiology, the myosin is pulling on the actin, shortening the sarcomere, shortening the Z to Z distance. Okay, so let's skip this. Certain part, what's not changing, the length of the A band is not changing, because that's defined by the length of the myosin filament. What's changing is the space between the I band and the H zone. Okay, so I'm going to let you do this one as homework. Put it in order, right? This is a homework assignment to this is alphabetical order. Put it in correct order for describing the cycle of the cross-bridge cycle that defines contraction. So the answer is on Moodle, which I will skip, okay? But that's homework. Do that problem at home. You probably know that I like those kinds of questions, putting things in the right order to tell me a story. So practice it. So what we have just defined is a twitch. We have shortened the muscle by a twitch. You know what a twitch is, a twitch. We've shortened the muscle by a tiny amount. We summate that to do contraction. So for example, one action potential of the muscle causes one pulse of cytoplasmic calcium, causes one twitch of force. So this is voltage, this is calcium concentration in the cytoplasm, and this is some unit of force. And that's a twitch. A little pulse. This is 200 milliseconds, so a little pulse of contraction. That's the unit, right? Those are the units. Two action potentials, two summated pulses of calcium, two summated twitches. 4 action potentials, 4 summated pulses of calcium, 4 summated twitches. You see what I'm doing is I'm building up, I'm using temporal summation to build up force. So the way we control the force of a muscle, one of the 2 ways, there are 2 ways that we grade muscle contraction. We're not twitchy, we can move our muscles smoothly. I mean, we're twitchy sometimes, but we're not just always jerking around, we're moving our muscles smoothly. Because one of the ways is through temporal summation. Higher frequency of action potentials summates faster and gets to a constant calcium concentration, gets to a steady state calcium concentration and a steady state contraction. So that's one way. Slow frequency, less calcium, less contractile force. High frequency, more calcium, more contractile force. Temporal summation. The frequency of action potentials, driven by the frequency of the neuromuscular junction, controls contractile force. The other way, this is a repeat. The other way we grade contractile force is by engaging more motor units. More motor units, more fibers are activated, more contractile force. And we do both of these. So we could do temporal summation of one motor unit and recruitment of multiple motor units and temporal summation of those in order to turn this weak contraction into a stronger contraction. And that's grading of contractile force. And we learn how to do this. So for example, here's a lightweight, right? And I have learned how much force it takes for me to lift this. But if I put some weight in here, I have learned, now I know, I predict, my brain is working, I predict I'm going to need more action potential frequency and more muscle fibers, both temporal and spatial summation, in order to lift this. If I did the same movement as I did before, I would not be able to lift it. But I have learned I can lift that. And I'm doing the same muscle, but the muscles are more muscle fibers and they're having a higher frequency of action potentials. But you've probably experienced the trick, right? I'll be the subject, but I need an assistant. What I want the assistant to do is to make some noise here and either have the buckets in or not in the box, okay? So I'm gonna leave the room because I can hear the sound of you moving the buckets. So somebody come here, take the bucket. I'm just gonna turn my head and close my eyes, right? No, I'm gonna close my ears, close my eyes so I can't hear anything. Somebody come up here and either leave the buckets where they are or take them out and then cover the box. So I don't know if you take the buckets out, I'll keep my eyes closed. Okay? So somebody do that. Are you ready? Nope. Pat me on my shoulder when you're ready. Got it. Guide me to the box.
Forward.
Okay. So I think it's full. I had a bad prediction, right? I predicted I needed more force than I really needed. And so you've done this, right? If someone throws you a styrofoam brick, oh my God, it's just a styrofoam brick. It doesn't weigh anything, right? So your brain is telling your muscles how much force you need. Where are the buckets? Okay. Yeah, good. So that's recruitment of motor units, right? Very few motor units. Put the weights in. Many more motor units needed to lift the same object. Okay, the last thing we'll talk about for just a moment is energetics. Most of the energy that you get in your muscles, most of the ATP that your muscles get is from oxidative phosphorylation. And you remember that glycolysis, this is getting back to self-is, glycolysis, but also protein hydrolysis and fatty acid hydrolysis. All of those things feed into the electron transport chain and oxidative phosphorylation, production of ATP. Okay? So in self-is, you only learn that glucose produces the NADH and FADH2 that is necessary to run the electron transport chain. But now I'm telling you I'm not going into metabolism, but protein and fatty acid metabolism also produce NADH and produce high energy electrons that your electron transport chain uses to make ATP. Your muscles also express a protein called creatine, which can be a storage molecule for high energy phosphate. So ATP can come from creatine phosphate dephosphorylation, as well as from oxidative phosphorylation in muscle. Many of your other cells do not store phosphate, high energy phosphate, the way your muscles do. But we have a store of high energy phosphate, and we have a rich way of making ATP by oxidative phosphorylation. Okay, so thank you for staying a few extra minutes. I'll see some of you in lab this afternoon.